In this video lecture, we will discuss about the molecular and genomic methods. But in the previous lecture, we have discussed about the microscopic methods and cultural methods to assess the or for the assessment of the biodiversity. In this lecture, we will discuss about the molecular as well as genomic methods. Here, the microbial cell is composed of a variety of nucleic acids, proteins, lipids and carbohydrates. Here the simple or single cell can contain or which contain the macromolecules such as nucleic acids, proteins, carbohydrates and lipids. As this all macromolecules, some are the informational and some are the non-informational. Here the nucleic acids are the informational data which is used for the assessment of molecular as well as genomic methods. This molecule have fixed structures. This have the fixed structure in a single cell and which can indicate a specific function which will generate and maintain the different lineages of microorganisms. What are the lineages? The species which has been derived from the its ancestors. What are the meaning of lineages? Lineages means the species which has been generated from its ancestors. Here the methods are designed to evaluate. What are this method? Why are this method designed? To evaluate the relation among the molecules which are directly relevant to recognize the diversity. Why are this method been designed or been evolved? To evaluate to me to know the relationship between the molecules which are nucleic acids, proteins, lipids, carbohydrates. All these are directly relevant to recognition of the species. These macromolecules are directly relevant or directly connected to recognition of diversity and the classification of newly discovered species. Why are these molecules used or have different function in the single cell or different cell, or these macromolecules varies from species to species? Here these are directly relevant or these are directly connected to know the diversity of any microorganisms. Nowadays, a molecular method along with the microscopic and cultivation method have been used to study the microbial diversity. Here, the molecular methods are also been involved into for the assessment of the microbial diversity. Now, what are the molecular context of microbial diversity? From which context it can assess the microbial diversity? Microbial approach is the most powerful tool because these nucleic acid sequence or nucleic acid sequence, DNA and RNA sequence is being specific for a single cell. It varies from cell to cell or species to species. The single sequence of DNA is been vary from single cell to the another cells for assessing the microbial physiology and genetics at the single microbial species level. Here the molecular approach is a powerful tool. How it is a powerful tool? The molecular means it can relate it from the nucleic acids which can differ from or it can vary from cell to cell or species to species. Advances in the molecular tools for studying the microbial diversity has been used for the analysis of nucleic acid, proteins and lipids. As I have previously said, the DNA sequence is very specific for a single cell. It cannot be similar or same for the another cell. It can vary from one cell to the another cell. Here the study of nucleic acid and proteins has been used. Here the study of nucleic acid as well as protein has been used to reconstruct the evolutionary distances and phylogenetic relationship among the microorganisms. Why here protein has been taken from the molecular context? How can it be different? The amino proteins are made up of amino acids and amino acids have different sequence on which they can code the amino acid and they can be useful for the single cell. It can vary from species to know the phylogenetic relationship between the cell to cell or species to species. That has been result into the development of the molecular clock. What is the molecular clock here means? The molecular clock indicates that it can be change or it can indicate the mutation rate from the over time. Here the mutation rate has been indicates from over time species, from one species which has been generated and from the another species has been generated from the previous species. The mutation, how much rate of mutation or how does mutation occur, in which way the mutation has been occurred from one species to another species, which can be referred as an molecular clock. That has been used for the phylogenetic analysis. The molecular clock has been used for the phylogenetic analysis of several groups of organisms, includes archaea, algae, fungi, bacteria and as well as protozoa. What is molecular clock? Molecular clock is an indicates or which indicates the mutation rate or how much mutation or which type of mutation is occurred from the one species which has been generated from the one species. That indicates the molecular clock. The molecular differences are fundamental determinants. Determinants means which can determine the differences from one era to from one species to the another species. Determinants of speciation and niche diversity. Niche means one area, the area diversity which can be in the prokaryotic population. This is all about the molecular context. What are the molecular contexts? The molecular context which involves the nucleic acids, proteins, lipids, carbohydrates, which can vary from one species to the another species. First of all, which can be determined or which has been priority to determine the molecular context, or which can be firstly used to assess the molecular diversity, which is an nucleic acid, DNA or RNA. The DNA and RNA sequence are very specific from one cell to the another cell, so it can vary or it can determine the assessment or which can be assessed, diversity can be assessed. The another one is an proteins. The proteins which have been through the proteins are made up of amino acids. The different amino acid codes for the different proteins. So there are specifically amino acids which can be determined or which can assess the microbial diversity. Here molecular clock is used to determine or to evaluate the mutation rate, or it can check the mutation rate from how the species to species mutation has been occurred. This is all for the molecular context of molecular diversity. Molecular context to assess the microbial diversity.